Hi, everybody. Welcome to another Chats with Chaudhry. It's a sunny day here in Surrey, and I'm delighted to be joined by Johannes Delp, who is the project leader for endotoxin services at Microcoat Biotechnologies. Today, we are going to be talking about low endotoxin recovery effect and why it is relevant in the first place, as it is one of the topics being covered at the upcoming Concept Heidelberg Pharmalab virtual conference, which runs from November the 21st to the 26th. And endotoxin testing is a huge part of the conference. And Microcoat Biotechnologies are actually speaking at the event. Hence, Johannes is here today. So, Johannes, lovely to see you. How are you?
Thank you for the introduction. I'm fine. How are you?
I'm very well, thank you very much. So, Johannes, where are you calling in from?
I'm calling in from Benried, which is actually a very small village south of Munich.
Well, I have to say then, given it's a small village in Germany, the internet connection is very good. So touch wood, it'll stay this way as we carry on our conversation. So I know Germany has a bit of up and down on the internet sometimes. So Johannes, before we start talking about LER effects, would you mind giving viewers a very quick overview on your own background and about Microcoat Technologies?
Sure. So I've studied regular studies in biology. And then very soon I figured out that I want to move into the biochemistry, biomedical directions. So I quickly focused on that. And then since my master's thesis onwards, I basically worked in toxicology. And I did mostly in vitro toxicology, focusing on neurotoxicology in the beginning. So for my PhD and also my postdoc time. And since 1.5 years now, I'm here at Microcoat and working in endotoxin research.
Yeah. Right. And what do Microcoat Biotechnologies do?
So a Microcoat focuses on a broad range of diagnostic services. We're a contract researcher for a lot of the big pharma companies and also smaller pharma companies. And we do a broad range of diagnostic tests for all different stages of the pharma production, clinical stages, preclinical stages.
Fine.
And we also have the endotoxin testing, which is then also full spectrum testing, so from developmental testing until release testing.
Right. Now, for those people who are not familiar with endotoxin testing, a really basic question: why is it important to do endotoxin detection in the first place?
So endotoxins are basically parts of bacteria, of bacterial contaminations. And for all kind of drugs that are administered parenterally, you need to check that whether you have a bacterial infection, contamination in the sample or not, because this would cause from fever to septic shock, a broad range of adverse effects in the patient.
Right. Now, I know there are a number of different methods that people can use for endotoxin testing. So would you mind giving viewers a quick overview of the methods out there and actually what the main differences are between each of the methods?
So there are, broadly speaking, two different ways of analyzing endotoxin. The most established method is, so at least up to now, is based on horseshoe crabs, which you then harvest at the coastal areas. You bleed them, and from their blood, you can derive basically the source for your assay lysates, and that you use then in endotoxin detection. And based on this principle, also recombinant tests have emerged, which use the same assay principle, the same enzymes in the cascade, or also truncated cascade, but it's fully recombinant. So you get rid of ethical issues by using these animals, and also you get rid of political issues or dependencies on only a few countries that can harvest these crabs.
Right.
So you become a lot more flexible and independent. And the second big principle is a human-based assay, which is based on either whole blood or peripheral cells in the blood. And this assay is then not only focusing on endotoxins, but is able to detect all different kinds of pyrogens. So all substances that would cause fever in a human being.
Right. Okay. Now, what is a low endotoxin recovery effect and why is it relevant and important?
So low endotoxin recovery basically describes that although you have your endotoxin present in your sample and it's not degraded at all, you are not able to quantify it by the test method that you use.
Right.
The formal definition is that you have 2 consecutive time points in a kinetic run where you have less than 50% of your expected amount measured, basically.
Right. Okay.
And the relevance of this effect is basically that if you do not test for this LER effect, you would perform your release testing, you don't find any endotoxin, and you would say, okay, this drug product is safe, I can administer it to a patient.
Right.
But you actually don't know it. And that's the reason why you need to check whether you have an LER effect or not.
Right. Okay.
And if you administer a drug product to a patient which has endotoxins present, this could cause a fever, septic shock, but it also could cause immunization of the patient against the drug product. And that's also something that you really don't want to have in
Sure,
treatment.
definitely. Right. So at what steps in the sort of pharma production process are LER studies required?
So there are 2 different studies which investigate, or 2 different study types which investigate whether you have low endotoxin recovery. So one of them is during the production process based on the in-process controls,
right,
which usually drawn from the production process, then stored at a given temperature and analyzed at a later time point when the QC lab has a free slot for the analysis. And for these samples, you need to determine whether this or how long the storage time can be so that you find endotoxin if it was present. And the second and maybe even more important test for LER is the studies which are actually called LER studies and which are regulated by the technical report 82 by the PDA. And these focus on the final drug product. And you basically have to show that if during the production process you had an endotoxin contamination, you would be able to detect it in the end when you do the release testing for the final product.
Okay. And how do you actually discover an LER effect?
So what you do to discover is you basically take your sample and spike a known amount of endotoxin into the sample, so that you know what the basis of expectation is, and then you incubate it for a given time, which is oriented on the production process, on the storage conditions, and then at different time points within this kinetic run, you take a sample and do the analysis of the endotoxin. And as soon as you find less than 50% endotoxin in 2 consecutive measurements, then you have basically detected an LER effect.
Right. Okay. Now, as I said at the beginning, Microcoat are presenting or speaking at the event at Pharmalab, which runs 21st to the 26th. So what are you speaking about?
So my colleague Michael Kraklau is presenting some of the latest research and development projects that we have performed during the last year or years, and will give some insights on how to discover LER effects, but also on what you can do when you actually have found an LER effect.
Right. Okay. So there you go, people. There's a quick overview on the LER effect. You can find out more about that and lots of other subjects around endotoxin testing by registering for Pharmalab, which runs from Monday the 21st to Friday the 26th, where there's lots and lots of great speakers, including Microcoat, as we just mentioned, talking about Pharmalab. I mean, Johannes, have you been to Pharmalab before yourself?
Yeah, I've been to the online conference last year.
And how have you found it as a visitor then going to a conference like Pharmalab?
Well, it was, of course, very informative. There are even parallel sessions. You have to decide which of the talks is most interesting for you at that given slot. But it's really a broad range that's covered by Pharmalab. And it's not only speakers from one branch. So it's big pharma is present, academia is present, smaller companies like ours are present. There's also some people from academia, some suppliers also are there. So we have really all the people that should be getting together are getting together.
Oh, well, there you go, people. So if you've never been to Pharmalab Virtual, I've been to the virtual event, but I've also had the pleasure of going to the live event a couple of times as well. And it's a shame it isn't live, but it is a very good event when you go to it live as well. But the virtual event will be very good. It's running over 5 days. There's lots and lots of talks and presentations, and they're split up into specific tracks per day. So endotoxin, if I remember correctly, is in the last 2 days of the event on the Thursday and Friday. But you can sign up for Pharmalab. The registration and the link to registration, more details about the event, is actually above the video. So, Johannes, thank you very much for taking the time to talk to me. It's been lovely to meet you.
Thank you too. Have a nice day.
Thank you. And thank you, viewers, for watching. If you want to know more about LER, endotoxin testing, or the different issues around microbiology, then sign up for Pharmalab. The link is above the video. And if you've got any questions for what Johannes has said today, then put them below the video. I'm sure Johannes will be only too happy to answer any questions you've got. So until next time, as always, thank you, Johannes, and thank you for watching, and stay well and stay safe. Bye-bye.
Bye-bye. Thanks.